So, we'd like to hear from you what you'd like to do. Shall we do this talk about the basics and then talk? I see people nodding heads. We're getting more people, aren't we? Can we do that? Everybody, there's a couple handouts here. One right here. If you haven't gotten one, maybe you can take that. I'll be as quick and kind of summarize it as best I can. The handout has an article that goes through this again if I lose you in a few spots. You can read it again in the article, and then there's references there. And the issues that arise from genetic engineering are listed there. But I'm not going to go into the issues because there just isn't time. If there's any question about the effects of Roundup, because Roundup is used in genetic engineering, I have a file here of the health effects of Roundup studies. These are scientific studies. The health effects of Roundup and the soil and plant effects of Roundup. Their studies are ancient, but they're listed on the front because Monsanto promotes it as being environmentally benign, and I find that offensive. Everybody look at your hand. Okay, what do you see there in your hand? Tell me what you see. Bones?
Skin.
Skin?
Creases. Lines.
Muscles? You know there's blood in there? Hair? Nails? The things you are made of. All those things that we mentioned that you're made of, that you're looking at, what are they made of?
Cells.
What's your skin made of?
Water.
There's water in there. Proteins. They're all proteins. Basically, there's two kinds of proteins. There's your structural proteins, and there's another kind of protein called enzymes. Before I started studying this, I thought enzymes were the things that digested food in my stomach. But I'd like you to expand your concept of the enzymes, because enzymes, there's a whole bunch of them, and they're in every cell of your body, and they basically are guiding all the chemical processes going on in your cells. And everything happens in your body coming from a cell. Muscles contract because there's cell muscles contracting. Nerves do their sending of messages because there's a cell doing the chemical exchanges that cause those signals. Everything in your body works starting from a cell. And that's what gives us an understanding of genetic engineering. So we start with a cell. And I have a metaphor here that kind of gives us a mental image. You really don't have to know the words, but you can see the picture. And the metaphor is to think of a cell, and this can be both an animal or a plant cell, as a medieval city. Because a medieval city had, remember, it had a city wall, and cells have membranes that have gates in them. Medieval cities had gates in them. And that's what a cell has, and only certain compounds can get through the gates. A medieval city had a castle, and the cell has a nucleus, the center of information and direction for the city. The ribosomes, what in a cell is called a ribosome, are the manufacturing plants, and they're distributed throughout the cell, or throughout the city. There are recycling plants in a city, and also in a cell, where compounds are broken down and recycled. There's storage, food storage in grocery stores in a cell. There are a whole network of membranes for the roads and bridges. There are guards at the gates who determine what compounds go in and out. in biology they're called receptors. There's post office that delivers messages. There's a whole system of messages, a whole system of post office carriers, message carriers, that are specific enzymes or compounds. We'll get more to enzymes in a minute. So think of the complexity of a city. Oh, and well, the enzymes are the people doing all these jobs. And just like a city, each enzyme has its own particular job. Just like in a city there's a postal worker and a taxi driver and a cement mixer and a construction worker and the farmer. Each enzyme, which is a particular compound, has its own job, and that's the only job it does. And an enzyme is a protein, and there's thousands of them in your body doing thousands of different jobs. You have liver cells, you have muscle cells, you have heart cells, and in all of those cells are different enzymes doing different jobs. So each a group of cells is unique. So with that metaphor in mind, let's go to how a protein is made. Because we're getting toward how genetic engineering is done. So in the castle, in the nucleus of the cell, in the castle of the city, is the source of information and the instructions, the DNA. And you probably know it's like a twisted ladder in shape. It's a long molecule that has the repeating compounds, 2 rows of them with cross bars, sort of, and it's twisted. Double helix is called. And in one cell, if all the DNA could be stretched out in one line, in one cell, it would be 2 meters long. That's how much it is. But it's all crimped up and put together in the nucleus. In its form is the crossbars of the ladder have specific compounds on the outer edge of that ladder step. And there's only 4 different ones in DNA. And they're given names, scientific names of course. And the first letter of the name is kind of what they go by. And there's only 4, and they are A, T, G, and C. And those are called bases. Those are the bases. So when you hear that term, you'll know, like when you hear the term sequencing of DNA, like when they sequence the human genome, that means they've read the order of those 4 letters through that huge, huge, huge compound. How many? 3 billion bases in the human genome? Your handout has the numbers on the chart of how many bases there are in various organisms. So it's just 4 letters, and the order they're in that is this genetic code. Well, how does that work? First of all, we know that DNA makes proteins. And proteins are what, either in the form of building material or enzymes, proteins are what make all of life work. And all of life is built on it. That's the only thing DNA makes, is proteins. Either the building or the enzyme parts. So how does it do it? This is kind of a breakdown of what happens in the DNA. First of all, a signal comes in maybe from outside the cell, maybe from outside the organism, or within the organism. A signal comes in, and there's also receptors around the nucleus, and the signal comes in that says it's time to make a protein. So there is a complex enzyme, this red oval refers to that starts the process. It comes and attaches onto the DNA, and it attaches at a particular site that is called a promoter, which is just a particular order of those 4 bases. And then it goes, it moves along the DNA, it opens it up, and it copies one side of the DNA. While it's doing that, other enzymes, jobs, people, are bringing materials to it, the materials it needs to make a copy. So it copies it out and say, here's the first copy that it did after it went along, and it actually stops at a terminator spot. So if you hear that term. And during the process, only one thing is changed. The T is changed to a U. So we still just have 4 bases, and we only have 1 line of the molecule. And it's a stretch of what was in that 2-meter long DNA. And then there are other enzymes of particular jobs that come along, and this one's called RNase, and its job is to cut out the parts of this that don't really relate to the protein that this is trying to be made. Because the astounding thing is, in us, only 1% of our DNA actually codes, and I'll get to that in a second, for protein. 99% of our DNA is other stuff of our DNA at our level. Lower down in the level of complexity of living organisms on the earth, there's less other stuff in the DNA. So as for bacteria, for instance, more of their DNA actually codes for the proteins that they make. But in us, only 1% of that 2-meter long molecule codes for proteins. So there's a whole bunch of other stuff. And the parts that code for the proteins can be in segments interspersed with other stuff. So this enzyme's job in the city is to cut out what's not needed and paste together what's left. And it does that. There are other enzymes that can change the order of these letters, of the bases, in every cell. And there are signals that guide the spliceosome that tell it what to do in its splicing and cutting and pasting, kind of like your computer cuts. You cut and paste, you know. Okay, so then after these enzymes are done with this product, you have what's called the messenger RNA, which carries the code of the letters, the order of the letters, for this protein. And that all happens in the castle of the medieval city in the nucleus. And then the taxi drivers of the city, the enzymes that are the taxi drivers, come along and pick up the messenger RNA and carry it out of the nucleus, out of the castle, to the manufacturing plants out in the cell, where other enzymes are bringing the materials, like from the recycling plant, to the manufacturing plant, the ribosomes, and getting ready to make a new protein. So the taxi drivers take it there, and then the enzymes start bringing the material, and there's a special enzyme that now translates this code of bases that is the messenger RNA to what? Does anybody know? What a protein actually is made of is a string, yes, a string of amino acids. It's like a string of beads. And how many amino acids are there? 20.
28.
20. Less.
20 that are used.
Huh?
Anyway, okay.
Okay. And does anybody know what the code is from the order of bases to the amino acids? How many base letters, letters representing a base, give a particular amino acid? Right. and this is the amazing thing. This is true for pretty much all of life on Earth, that a specific order of the bases gives a specific amino acid of the 20 amino acids. So the example I have here is UGG, UG, translates, when the enzymes come along and do the translation, to the amino acid tryptophan. And then, so the order of amino acids determines the function of the protein, or which protein is being made here. So, you got 20 amino acids, you can have a whole bunch of combinations of amino acids, giving you a limitless number of proteins potentially. Proteins average between 40 and 1,000 amino acids. And their arrangement in linear arrangement first. And then there are enzymes whose particular job, the only job they do, is to clean this string of beads up, clean the aminos, clean it up, because there can be mistakes or incomplete jobs. So it comes along and cleans it up. There's other enzymes that, well, I guess next would be the enzymes that add elements. There are elements added to this, like copper, sulfate, some of the elements of the, of the periodic table, or vitamins are sometimes added. And then the last step in the production of a protein is the folding. And it's folded in a particular way. And that also is according to what has been added to it and what amino acids are in there. Because some amino acids have an affinity to associate with others in a certain way. So where they are in the string determines how that string of amino acids folds into a three-dimensional folded molecule. And that three-dimensional folded molecule determines the function of that protein in the body, in any living organism. I'll just remind you how plants reproduce. Plants have flowers that give pollen, and the pollen has to find its way to the stigma of the flower, and then it grows a pollen tube down to the ovary of the flower. And there are so many defense mechanisms in plants that only within species can this occur. So in the taxonomy defines what we humans have. We formed a method of identifying plants: kingdom, phylum, class, order, family, genus, species, right? So that's our method of identifying plants. That determines different forms of plants. And that also tells us which plants cross-pollinate in nature. Because if they couldn't cross-pollinate, the ones that do cross-pollinate have a similar form. If they can't cross-pollinate, then they have differences in form. And that determines how we classify them. So only within species is this process working. There are defense mechanisms. There are also defense mechanisms in a cell to not allow foreign material in, and to break it down or silence it when it does come in. So what do we need to get over those defense mechanisms in the cell? First of all, we need something that carries our DNA material into the cell and into the nucleus. And what's used for that is, what invades organisms?
Virus.
Pathological organisms, bacteria and viruses that cause diseases. They are the things that have the skill, the chemical skill, to invade. So that's what's used in plant genetic engineering is a very skillful, actually it's bacteria, Agrobacteria tumefaciens causes tumors in plants. And so they take the DNA from that bacteria and use it, the part of it that is the invader, to invade the cells. And then you need the promoter, that is the bit of DNA that tells this enzyme to start the transcribing. And that's taken from cauliflower mosaic virus. And it's a very promiscuous, strong promoter. It causes if this is inserted successfully, it causes it to be expressed at a level beyond the regulatory mechanisms of the cell. It's a very powerful promoter, called a promiscuous promoter. And also, cauliflower mosaic virus is what was found in the corn in Mexico to identify it as being contaminated by genetic engineering materials. That's what they found. They didn't find much of the other construct. And then you need a way to know if it's successfully in there. So you add to your construct the DNA for antibiotic resistance. So you put this all together in a beaker, and you can do that if you isolate it here and then pour it in with some other DNA. DNA is kind of like Velcro. It has a hook and a loop on the end of the molecule. So one hook and loop joins up with another hook and loop, and then continues on. That's kind of a metaphor. But that's how they put it all together. They isolate each one, some of this and some of this and some of this, pour it together, and it hooks up on its own. So then they have these constructs. So you have the vector, the promoter, the marker. And the way the marker works, you squirt this into the petri dish, and the promoter, or the vector, carries it into the cell. The promoter gets it operating, and then the trait is operating. The terminator is just what stops the processing of the DNA. So then you have it in there. You squirt the petri dish with a little bit of this antibiotic. If the cell is expressing antibiotic resistance, it will survive, which tells you that that was a successful insertion in that cell. And so maybe 1% of these cells will survive. And those are the ones where it all worked. You got in there and they're expressing. And so you grow that 1% out and see what kind of potatoes you get. And then you go from there. And then you start reproducing that. It's clear, it should be clear hopefully. There's another method of, instead of using the vector, there's something called a gene gun where they coat tiny particles of some metal and with the construct and just shoot it into the cells and hope it hits in the right spot and is incorporated into the DNA properly. So, as you can see, there's no control over where in the genome the construct lands. There's no control over how many copies in the cell you're going to get. There's no control over what effect it has on the DNA of the host cell. Or it can also scramble the construct. It can scramble the host cell DNA. It can scramble the construct. Disrupt what's already there. There's no control over whether the plant defenses are activated or not. There is defenses, as I said, in the nucleus to break down or silence foreign material. And then, even if it is in there, if it's taken, the BT trait is taken from bacteria and put into potato, there are even further crosses from animals to plants that I'm sure you've heard of further away in the, in the tree of life. There's no control over what happens between here and here. If it's in an animal DNA in a plant cell, what do all these enzymes think of that? What are they going to do with it? How is it going to end up in the final shape of the protein? So it's inherently unpredictable and dangerous by means of these two processes. One called genome disruption, because it's disrupting the host genome. And the other, horizontal gene transfer, relates to the fact that bacteria and viruses don't really need to mate to transfer DNA. They can just sidle up side by side and say, "Ooh, you've got something nice over there. Give me some of that." And the other guy says, "You got something interesting over there. Give me some of that." It's called horizontal gene transfer. Bacteria do that. Bacteria and viruses do that. It's well known because that's why we have all this, these diseases that are now antibiotic resistance in medicine, because the bacteria are transferring the antibiotic resistance. So via horizontal gene transfer, because we are using pathological organisms, we are increasing the potential for, well, look at your list of concerns in the handout, for all kinds of things to happen as a result of further stimulating horizontal gene transfer and the things that can result from that. I'm going to stop there. We've got this thing called substantial equivalence, which we really need to talk about, because that's what all the regulation in the United States is about. And basically it says genetic engineered crops are no different than traditionally bred crops before genetic engineering comes along. And then the Europeans are promoting the precautionary principle and the biosafety protocol. Those things are important in this discussion, but I think I've taken enough time and I'm going to sit down. I hope that helps. I hope I didn't lose you too much.
Questions or? I mean, what happens here, I mean, there's two big myths that the corporations are foisting on the rest of the world, and very systematic. You heard the guy on the TV here talking about public relations, and you know, this is a very concerted $50 million a year campaign to promote two myths. One is that this process is safe, predictable, well understood, and just like conventional plant breeding, and it's highly regulated. Well, it's obviously not. We don't have a clue, is what it comes down to, and there's a number of examples of that are out functioning in nature, and Monsanto's even been sued over a couple of them. And the second big myth is that this is kind of risky, but you hear, you know, we got to have this technology to feed the world, and there's no alternative. There's no other way to feed the burgeoning population of the planet. And so these are the two that I run into all the time. Every time we have some debate with one of these guys in industry, that's what you know. They're in the vernacular of public relations. They're on message, and boy, are they! I mean, every time you hear this, it's the same thing. This is really predictable, well understood, and highly regulated. Well, if anybody wants to read about what Jill's explained here in more detail, the February 2001, is that what it is? Harper's, actually has an article by Barry Commoner in it that explains, you know, in written form what Jill's just explained, if you want to review this. Very clear, semi-technical explanation of what we don't know. That this whole system rests on the assumption that one piece of DNA makes one and only one protein. That if you put that piece of DNA from a bacteria, that construct in this plant, and that's the only thing it's going to make. Well, that's wrong. That's flat not true. I mean, you can get, there's a fruit fly that they've done work with where one string of DNA has been proven to make 3,000 different proteins, depending on environmental responses, all these triggers, all these multiple mechanisms in there, how you amplify, And there's no way to predict that control and both in the cell and the response to the environment that triggers those kind of changes. That's something we don't even begin to understand, what regulates those expressions. And an example of that is in Roundup Ready soybeans. They've taken a gene, this construct right here, to make an enzyme that breaks down the molecule that's toxic to plant that makes Roundup a good herbicide. Roundup kills everything. If it's a plant, it kills it. And so this makes the plant, Roundup Ready wheat, will have a gene that makes an enzyme that breaks down glyphosate, the molecule in Roundup, before the plant is killed. And that's what makes it resistant. Well, they've done this with soybeans. It's grown all over the place. And that theoretically is the only thing that gene is doing in that soybean plant, right? Well, what happened is, summer before last, in most of the South, they had a real heat wave. It was Alabama, Georgia, and all of a sudden, all this genetically modified soybeans, the stems started shattering and fell over. And this kind of upset farmers, obviously. And non-genetically engineered soybeans of the same genetic types, varieties, didn't do this. And so what happened, sticking that construct in a soybean plant, made it resistant to Roundup, but it also changed the architecture of the stem. That's not one gene happening to one spot in one plant. That's a fundamental alteration of the architecture of lignin and cellulose metabolism in a plant, and probably it's 50 or 60 different functional enzymes that were affected to go into making a stem. That's how little we know about this system. These guys are clueless, and they sit around and tell us that this is all heavily regulated, and it's not. Roundup Ready wheat will be introduced in Montana with no publicly available safety testing whatsoever at all. FDA just says, well, it's the same as regular wheat except for that one gene, go ahead and plant it. The regulatory approval for Roundup Ready wheat will be a variety permit issued by USDA, and they issue a variety permit for every other kind of variety that's released in general commerce. There is no safety testing, no evaluation of this, no understanding what's going to happen when they put that out there, when we eat it. And
It's out there right now, right?
it's in 30 some odd different field trials in Montana, and it's more in the Dakotas, yeah.
Well, I've heard so much about gene splicing, surgery, where they put one gene just where they want it on the chromosome. Does that exist? And Jill's talking about something else?
No.
Or they're kind of beautifying this process?
They're beautifying it.
Like
This
a
is a
smart
complete-
bomb that hits the precise target?
Yeah, that's a very good analogy. This is more like a cluster bomb that hopefully one hits. Yeah, there is no precision or control. They literally, a gene gun is a little tube full of gold balls, a couple of microns in size, coated with DNA, and there's a little .22 caliber cartridge behind it. They whap it and it blasts it in that petri dish. That's how it's done. And that's how Roundup Ready wheat was made. And it's not, it's not, genetic engineering. It makes it sound just like that, surgery.
It's not surgery, really.
No, it's. So this is, and there are many examples coming out, like the one of soybeans. I remember that because I talked to a soybean farmer who was really fairly upset about that. But there's many examples where the genes have unpredictable effects in the plants where you put them, and are having unpredictable effects in plants where we didn't put them, which is this horizontal gene transfer. This stuff, I know the USDA scientists in Iowa who work for EPA trying to argue about how big a buffer strip you should have around a cornfield to prevent the transmission of corn pollen and contaminate your neighbor's corn if they didn't want to be GM. We're talking about it should be 200 yards or half a mile or whatever. Well, as Jill said, cauliflower mosaic virus from genetically modified corn in Iowa is in ancestral land races of corn in Oaxaca, Mexico. That's about 2,300 miles last time I looked at a map. So the 1 mile we were fighting about really wasn't going to cut it. And we have so little understanding of even the social and economic mechanisms that transfer genetic material around the world that nobody in the entire U.S. government arguing about this thought to think that some Mexican peasant that got given a bag of corn might save some seed and plant it. That's how little we understand the social structures that go with this. And you know, but so remember this the next time they're saying this is safe, it's predictable, it's highly regulated. They don't have any idea how it's regulated in the plant where they put it, let alone wherever it might end up. Some British scientists have found antibiotic-resistant genes from genetically modified soybeans, that marker there, which is really what scares a lot of geneticists and biologists, is the antibiotic resistance markers. They found those genes from soybeans in bacteria in human guts. In E. coli that lived naturally in the intestines, there was horizontal transfer of that genetic bits of information. And having antibiotic-resistant genes floating around in normal human flora is not a really good thing to do.
This horizontal gene transfer, the colon and the gut are bioreactors. They're the
Yeah.
perfect place. This is where this horizontal transfer is taking place, in the dirt, in these different kinds of bioreactors. Just to touch on the history, the central dogma, 57, Crick and Watson, it was the observation of horizontal gene transfer and how the phenomenon takes place with bacteria and viruses that allowed people to understand the composites and what we call genetic engineering. We saw it happen naturally in the 60s and 70s. Said, wait a minute, this is simple, we can do this. We decided to play God with this biological phenomenon that's going on since time immemorial for 20 or 30 years now, and we feel we understand it, we feel we can utilize it for whatever means. We observe the phenomenon in nature, now we're duplicating it as a caveman did with a catapult.
Except this is a sort of self-reproducing catapult. Yeah.
Jill said that in Mexico they found the promoter,
Cauliflower mosaic virus?
yeah, they found that in the corn.
Yes.
Did that separate itself from the gene they intended to put in it? Or were both, do they stay, if one's there, they're both there?
It was, the construct was broken up, and their testing was able to detect cauliflower mosaic virus the easiest. So that's what they detected.
They don't really know that the unwanted gene made it into the corn?
Yeah, well, it had to have made it in to have the cauliflower mosaic virus
Okay.
there as part of
But they can separate, one's not necessarily an indicator that the other is there.
Well,
It is.
what they do is just, they look for cauliflower mosaic virus because there's just easy techniques to find that sequence of DNA. There's other pieces of DNA that they can label with fluoresce and things, and it sticks on that piece. So they found that and reported that because that's the technique. It's the easiest way to find foreign DNA is to look for that particular promoter sequence because there's ready assays. That's how, when the Japanese or the Europeans are looking at at imported crops from the U.S. and they want to decide whether it's genetically modified or not. This is the test they do, is to look for that sequence of DNA from the cauliflower mosaic virus promoter, because that's the promoter that was used in all the commercial crops so far has been that promoter, and they just have a test to find it. That's why they did it.
But it is an indication that the unwanted trait made it in also.
Oh, yeah.
So that's what I wanted to know.
Yeah, that has to have made it in there to be expressed, yeah. And that particular piece of research brought the wrath of Monsanto down upon a guy named Chapella, who was a professor at UC Berkeley who found that. And they threatened to withhold money from UC Berkeley, and he lost his tenure.
Did he lose his tenure?
Yes, he did. Yeah.
What was his name?
Ignacio Chapella, really good geneticist. He had that paper published in Nature, which is probably the single most prestigious scientific journal on the planet, and Monsanto came down like a ton of bricks on Nature magazine over that, publishing that article.
Well, has there been any retraction to it, or I mean?
Yeah.
Now, what there has been is confirmation by 3 independent Mexican scientists from CIMMYT, the big Mexican grain research facility, and they've gone out and looked and they found exactly what he found.
And how do they justify Quinn's tenure?
They didn't. They just did it.
Most of the universities in the United States are going to biotech.
That's where all the money is, right?
That's where the money is. I mean, it's a gold rush. It's a big gold rush. That's what's driving it really is money and venture capitalism. And now it's also dependent upon the Bayh-Dole Act, which was passed in 1980, which allows universities to profit from their research. And so now the universities are and the corporations are working together, forming contracts, whatever, to profit from their.
What was that actually called?
The B-A-Y-H Dole, Bob Dole Act, passed in 1980, allowing universities to profit. And one of the things we need to work on is to revoke that act, because the universities, the land-grant university charter was to give free, to do agri-research and give the results freely to the farmers in each region. And there was a land-grant university in each state that was chartered to help the farmers for free. And they did their research based on federal money, tax money, but it was for the farmers in their region for free. Now they're all selling out, all except Steve Jones in Pullman, Washington, who's the last wheat breeder who hasn't sold out. And boy, there's a lot of pressure on him, I tell you, to go. But all the universities are selling out and going into biotech. That's where the venture capitalism is.
Happening right here at Dixie University.
I bet.
Oh, it is, yeah, Monsanto was contracted with a guy named Luther Talbot here, who used to be a public wheat breeder, and he's now the private wheat breeder. He's doing the Roundup Ready wheat. Jim?
What year did Chappella discover the archival virus in the colon?
2001, I think.
I think that's.
I think the paper was published in early 2002.
And what Nature magazine, what article in Nature was that?
It's, I don't remember the title of it. You'd have to just go online and find, this, if you go on the Nature, Nature website and plug in Chappella, I'm sure you'll pull it up.
How do you spell that?
Chapela. Might be
Two L's. Two L's.
To follow up on my question, and maybe either one of you have seen this, but I saw somewhere recently, and I can't cite the source, that like maybe 8 or 10 Mexican states now have confirmed that they have contamination, and some of them, I think especially Oaxaca, they're very widely, I forget how many samples they did, but a very significant percentage of the native corn now is expressing this trait. It was discovered 2 years ago, and now it's all over the place in 2 years.
I saw that article too, yeah. I think it was 8 states. First, Chappella said found it in 1, maybe 2 states. But the article recently said
Do you know where you saw that, Jill?
Yeah, it's from RAFI, which is now called ECT organization. But it used to be called RAFI, R-A-F-I. They kind of sponsored the studies that, along with Mexican organizations that went to the other states to do the research. And they just sent that out on the web.
Can you explain to me then, in the Roundup Ready wheat that we know is being tested in the ag research stations in Montana and North Dakota, if that was grown there this summer, can the pollen from that cross over into neighboring fields or neighboring counties and contaminate the wheat we grow? As this corn has been?
First of all, let's understand that corn is an open pollinated plant, meaning the male pollen is up on top, the female silks are down below, the pollen, and it's wind, it's dependent on wind, it's wind spread. So pollen can go all over the place.
What about
wheat, on the other hand, is not an open pollinated plant, it self-pollinates. So the opportunity for cross-pollination is much less in wheat. However, the plant geneticists say it's around, I think, 5% potential of cross-pollination in wheat via insects, probably, mostly, maybe some wind. So I'm afraid the answer is yes.
But the other issue with wheat is again where we forget about the social institutions here that go on. Montana, the Dakotas, most of the wheat grown in the Dakotas and Montana is exported through Portland, Oregon to the Orient. And the wheat system, storage, harvest, storage, transportation is like a giant funnel. It all ends up in Portland, Oregon. And in that giant funnel of elevators and combines and trains and there is absolutely no capacity to segregate one kind of wheat
Yeah.
from another.
Yeah.
I mean, you know, it's, and the parallel infrastructure, you know, Monsanto's merrily said, well, we'll segregate it. We said, well, who's gonna pay for the parallel infrastructure? Well, boo, boo, boo. You know,
Their response is that farmers should have a choice.
yeah. Right, and so, you know, Montana farmers have a choice to build a new
Mike
Burlington
Malone
Northern,
with
no problem.
Monsanto, that's his mantra. Farmers should have a choice, and we'll work out the system, but farmers should have a choice.
Is that paper or plastic?
Yeah.
And I want to touch briefly on the brave new frontier of this stuff, because this is now that you see, you know, this, you think this is scary of wheat or corn, there's a bunch of small venture capital finance, mostly venture capital finance companies around the U.S., almost exclusively in the U.S. There's a couple in Europe, but the Europeans don't like this very much, that are using genetically engineered plants as little mini factories to produce human therapeutic proteins for medicine. That, you know, growing large quantities of human cells in tissue culture to produce a protein is pretty expensive. Tissue culture costs a lot of money. So the idea here is you can take a gene that might have some medicinal function in a person, in people, and you can take that gene out and stick it in a corn plant and make, voila, make lots of protein. They're doing this, the reason they made Dolly the sheep, remember Dolly the sheep, you have a cloned sheep? Well, they wanted a nice cloned sheep to make sure, you know, from a single cell, none of this messy sex, to mix up the genes, because they wanted to use sheep as a way to overexpress certain proteins in their milks for medicine kinds of stuff. And so there's people running around merrily putting human therapeutic proteins and animal vaccines in plants. And
Mostly corn.
Mostly corn. They use
Open
corn
farming.
because they understand the corn system. You can raise a corn plant from a cell pretty straightforward now. So they all use corn because it's cheap. And some people sort of raise their hand and say, well, corn's also the third most widely consumed crop on the planet, and maybe that's not the best choice as a crop, but they say, oh, hell with it, and do it anyway. And, I mean, to give you an example, and I brought this up a couple times, I had some scientists that not believe me, but there's a company in California, this is the true brave new frontier. The company's Epicite Corporation, San Diego, California. They have a name and address and a phone number and a website. And they are using corn, genetically engineer corn, to express a protein that's a human spermicide. I am not kidding. And they have a permit to grow this corn outdoors in a field.
Wow.
Granted by the USDA, who is currently controlled by a former Monsanto executive. But, you know, this is how little thought we give to, and they're arguing about whether the buffer zone around that field should be a mile or not. And I mean, you can read I read some of the arguments about whether that, you know, Mexico seems to have completely eluded these people. Mexico, you know, San Diego is not really all that far from Mexico if you think about it. And this is, you know, and they've done this and they put it in a field trial. And there's obvious stupidity of it. But the only driver for this is sort of scientific sexiness and money. There is no, what this is a protein that binds the sperm and causes the sperm to stop swimming. So it's just another vaginal cream. And so it's a biochemical condom. Well, you know, what's wrong with regular condoms? I mean, we're doing it for no real good, compelling reason, other than that this company thinks they can make more money at it, and without any thought to what may be going on. And they've done this with pig vaccines. Pigs are pretty close to people. And the company that did the pig vaccines, I think, has done 6 field trials, and they've lost control out of 4 of them. You know, this is not a real good record. They found corn from their field trial in contaminating soybeans. And we found a USDA inspector found it by mistake. And this is not some wild, highly sophisticated mechanism that transferred this stuff. It was, they rented a farmer's field, and one section of the field didn't work very well, and they plowed the corn down. Well, corn grows from seed. And so next year they planted some soybeans, conventional soybeans, and some of those corn seeds germinated, had the gene, and contaminated the soybeans. I mean, this is not rocket science to figure out where this contamination came from. And they can, with about half an acre of volunteer corn plants, they contaminated 500,000 bushels of human edible soybeans. And there's no way to contain this stuff. It cannot be done, no matter what they tell us. It's
Just by material being harvested with the soybean.
Now, was that horizontal genetic transfer?
No, that was
it's
just a combine.
Because these genes are expressed in every cell of the plant, so
Right.
even if you have a leaf of the corn, you know, that gets mixed up with the harvest of the soybean, you have those cells that have those genes in the material, and that's how it contaminates.
Yeah, this is
by
just volunteer
physical
corn that grew that next
Yeah,
season.
When you said that was a combine, you're not being, that's
No,
not.
I'm not.
that
They
was
just
a
combined the soybeans and chopped up the few corn plants in there and contaminated the whole lot. It wasn't.
With the combine.
So how much corn contaminated how much soybeans?
It was about half an acre of volunteer corn, and they contaminated roughly 500,000 bushels, if I remember my numbers right.
What was the contaminant?
It was an experimental protein that's basically it's a vaccine for a respiratory disease in swine, in pigs, yeah.
I want to be clear, you're not saying that the gene transferred to the soybean.
No,
Okay,
no.
you're saying that the bulk of the food was contaminated and anybody that ate it would be getting,
Some fragments of that corn. yeah.
okay.
And that may not sound like much, and in that case it might not have been, except that one of the real concerns that people have over this is making proteins, either the target protein, in this case a swine vaccine protein, or some other protein that you don't predict, that people are allergic to. Proteins are the most common allergens, and so even a fairly small amount of some weird protein, and the swine protein could be a good one, can trigger allergy in sensitive people. So you don't have any good way to predict that.
The next question would be, once discovered, what happened to the 500,000 bushels of soybeans?
They burned them, theoretically. That's what they said, they were going to be destroyed.
Yeah,
They
right.
probably use it for animal feed.
Yeah.
They may have, I don't know. But the other point is that this was an accidental routine inspection. And there's, one of the things that's been done here is a systematic reduction of agricultural inspectors to look for this sort of stuff. I mean, we have no idea, but this company in Texas really has a very poor, they've lost 2 or 3 other of their trials besides this one. This one just happened to make the news because there's a lot of soybeans.
I have a question that basically what you're saying here is that all of this genetic engineering is to make a product that will be sold and is by big corporations and makes a lot of money for big companies. What if you use this technique to put vitamin A in greater quantities in corn or rice or wheat? It wouldn't make money for anybody. It wouldn't sell. But what if you were able to, this is not something that people are doing because it hasn't a market.
Well, it
Nobody's
is something
going to do
people are
that.
doing. This is actually the cover of Time magazine. This is golden rice. They, I don't know, actually it was the Rockefeller Foundation who put up most of the money to engineer rice to produce beta-carotene, the precursor of vitamin A, to treat vitamin A deficiency blindness, which is a really terrible problem. I mean, this is
In the third one.
in the third world, particularly in Southeast Asia.
What issue of that was Time?
Yeah.
It's a couple years ago.
Oh, okay.
It's July 31, 2000. And this brings up, I mean, this gets to the next piece of the discussion here is big myth is we got to do stuff like, that golden rice is going to save kids from blindness, and we're going to, I mean, the latest one I read was we've engineered a potato to overproduce a protein, so now you have high protein potatoes in India. And all this sounds really technologically good, but it's primarily publicity stunts. I mean, they gave some money to this scientific team, and they solved some technical issues in getting there. But there is no commercial market for it, because the people who need vitamin A to make up for vitamin A deficiency have vitamin A deficiency because they can't pay for it, or as Van Danisheva said about I read some about an article she had about genetic, high protein potatoes for India. She said, well, what's wrong with lentils? You know, and the problem is people are too damn poor in India to buy lentils anymore. And so that's where you get into this much larger discussion of why people on the planet are really hungry. And maybe the Monsantos of the world are doing us a favor in triggering this discussion.